I'm Kathryn Beabout, and I'm a graduate student in Dr. Yousif Shamoo's lab. The topic of my research project is the evolutionary dynamics leading to tigecycline resistance in Enterococcus faecalis. So in our lab, we use a turbidostat, which is a fermenter-based system that maintains a large population of cells at its fastest growth rate. And we expose the bacteria to increasing drug concentration over time until we're able to select for drug resistance. And the advantage of this system is that we can track the evolutionary dynamics leading to drug resistance, so we can see when mutations that appeared at the end of adaptation first appeared within the population, and then what frequency they reached throughout adaptation. This research has implications in the clinical setting because we could potentially identify, for example, that certain types of bacteria with a certain genetic profile are more likely to adapt to resistance over other bacteria. And we could also potentially, for example, identify, like if there was a particular signaling pathway that was really important for drug resistance, potentially an inhibitor of that pathway could be used along with the antibiotic to make it more difficult for the bacteria to become resistant. This would potentially allow the antibiotic to be more successful during therapy and to maintain the efficacy of this antibiotic against these bacteria. So research is very challenging, but it's also very rewarding. So frequently I'll come up with a hypothesis and I'll test it, and the results won't be quite what I was expecting, so I have to change the hypothesis and redesign the experiment and try again. But eventually, over time, I start piecing together how the system works, and that's very exciting and rewarding because you're the first person to identify how this system works and how this molecular mechanism confers resistance, for example. So it's very rewarding.